This is USMLE QA, USMLE material presented in a question and answer format structured similar to the first aid for the USMLE Step 1 book.
Question: What are exons and introns in genetic information? Answer: Exons contain the actual genetic information coding for protein or functional RNA. Introns do not code for protein, but are important in regulation of gene expression. Question: How do exons and introns differ in their role in gene expression? Answer: Introns are intervening sequences and stay in the nucleus, whereas exons exit and are expressed. Question: How do exons and introns differ in their location in the cell? Answer: Introns are located within the nucleus, whereas exons are found outside of the nucleus and are expressed. Question: How does alternative splicing affect the number of unique proteins produced? Answer: Different exons are frequently combined by alternative splicing to produce a larger number of unique proteins. Question: Can you give an example of how alternative splicing can produce a variety of protein products from a single hnRNA sequence? Answer: Alternative splicing can produce a variety of protein products from a single hnRNA sequence. For example, transmembrane versus secreted Ig, tropomyosin variants in muscle, dopamine receptors in the brain, host defense evasion by tumor cells. Question: What is the function of introns in genetic regulation? Answer: Introns do not code for protein, but are important in regulation of gene expression. Question: How do introns differ in function compared to exons? Answer: Introns do not code for protein, while exons contain the actual genetic information coding for protein or functional RNA. Question: What is the role of introns in the production of unique proteins? Answer: Introns do not play a direct role in the production of unique proteins, but their presence in the regulation of gene expression can indirectly affect the final protein product. Question: What is the difference between introns and exons in terms of splicing? Answer: Introns are intervening sequences that are removed during the process of splicing, while exons are the sequences that are retained and used to code for protein or functional RNA. Question: How does alternative splicing use exons to create a variety of protein products? Answer: Alternative splicing uses different combinations of exons to produce a larger number of unique proteins. By using different exons and leaving out others, the final protein product can vary greatly. Question: Can you explain the process of alternative splicing in more detail? Answer: Alternative splicing is a process by which different combinations of exons are selected to be retained or excluded from the final hnRNA, leading to the production of multiple protein products from a single gene. This mechanism allows for greater diversity of protein function and regulation within a single genome. Question: How do introns influence the regulation of gene expression? Answer: Introns can influence the regulation of gene expression by affecting the process of alternative splicing, by providing regulatory elements such as enhancers and silencers, or by interacting with regulatory proteins. Question: In what ways can introns be important in the production of functional RNA? Answer: Introns can be important in the production of functional RNA by providing regulatory elements for the production or stability of specific non-coding RNAs, such as microRNAs. Question: Can you give an example of how introns can provide regulatory elements for the production of functional RNA? Answer: Introns can contain enhancer or silencer sequences which can regulate the transcription of the gene, thus affecting the production of functional RNA. For example, introns in the gene encoding for the pre-microRNA can contain sequences that regulate the production of the mature microRNA. Question: How does alternative splicing affect the variety of proteins produced? Answer: Alternative splicing allows for a single gene to produce multiple protein products by using different combinations of exons. This mechanism increases the variety of proteins produced and allows for greater diversity of function within a genome.
Thank you for watching our Q&A format video. We hope this helped you learn, review, and retain knowledge needed for the USMLEs. For more information on this topic, check out the link in the description. Now, if you want to take your USMLE prep to the next level, be sure to check out all our courses, which are structured to follow along with the first aid for the USMLE Step 1 book. You can find us at usmleqa.com. And if you're watching this on YouTube, be sure to like and subscribe.